genetic variation, and genetic marker. But before going to the content of today scientific point, I just want to inform you that I just received the information from the university this morning about from next week, our lecture course will be organized offline instead of online like today. So today is the last session that we will be organized online. And the interesting thing is, while all the lecture course, we have around 350, 400 students will be organized offline. All the other bioinformatics practical course or exercise tutorial course, which only around 30 or 40 students per class, will be organized online. So frankly, I don't really understand the logic behind this. So please don't ask me why the small class can study online, but the last class must study offline. I don't know. Anyway, maybe because that new normalization, and I know how you should feel, but me, I think that the lecture course should study online, should keep study online. The final exams should be organized offline, of course, because if the final exam was organized online, so if it's organized online, that means you are staying home and using the computer to do the final exam. That means if I give you a open question, that will be let's say the large proportion of the class will retake this course genetic. But if I give you a, let's say, a closed question, then a lot of you can use the internet connection to find a solution, and normally it's not a fair competitive. So I really think that the final examination should be organized offline. But in this condition, I think that the lecture course with 400 students be organized online. So especially because some of you are not located at Hanoi at the moment. So if you're going to study offline here, in Monday, in Hanoi, you will have to move here and waiting for accommodation, like rents and everything. And because the situation of the pandemic is more and more complicated, high school will changing the option from online again. So who knows? Maybe in 1 week more, we will be back to July, and I don't know. So anyway, I think because of the right. Wait a minute. Này thì mình sẽ nói vào việc cho rõ ràng. Thông báo là đi học offline tuần sau với đối với thuyết và buổi trong giấy theo mình thì mình cũng chưa hiểu được cái logic của trường. Và quyền hạn của mình thì không làm gì được vì có một điều chỉnh một chỗ như này để các bạn tiện cho cái việc học thôi. Đó là tuần sau thì bắt đầu từ 1 giờ chiều thì mình sẽ học một genetic thì thường là sẽ 2 lớp học, một lớp học từ 1 giờ đến 2 giờ 45, một lớp sẽ học từ 2 giờ 45 đến 4 giờ 45. Thì tuần sau để việc các bạn là đi lại
về cho các bạn học là một lớp lớp trong chiều chiều luôn 3 tiếng đồng hồ từ 11 giờ đến giờ 5 giờ và các bạn học lớp lớp 2 thì học buổi sẽ tiếp tục sau đó thì tuần đến thứ tư thì lại lớp 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 1 học. Đối với các bạn F0 F1 thì hiện giờ nhà trường đang đưa ra cái lớp Genetic này rồi học offline sẽ tổ chức theo hình hybrid, nghĩa là sẽ có một thuật liên hệ họ thu phát và là trực tiếp luôn luôn bay ra bạn nào F0 F1 có thể học được ở nhà mà không phải đi trường. Và mình đang gì với nhà trường và các bạn mà thuộc địa bàn Hà Nội nữa, nghĩa là không phải một bạn nào đang ở Hà Nội, các bạn ở tỉnh ngoài thì cho phép luôn những cái là học online trong những bạn và ngoài ra thì để tiện hơn các bạn trong cái việc học việc có thể một bạn không thể xem việc cái học là là nếu mà sao em nói hơi tình chưa thì mình xin hai hả một này vẫn chưa rồi hai cái này sẽ hỏi lại bên và gì mà đã được xem mà có thể có ai không. Thực ra mình muốn là muốn ở đây luôn lớp mà 350 người lấy nhiễm sẽ cao. Bạn thở là tổng thể là lớp là 110 sinh viên một lớp chia ra cao khoảng độ 300 sinh viên mình lấy nhiễm khá là cao. Nhưng mà hiện giờ những trường đi học lại trở lại thì mình chỉ là lao động thôi, thế là kỳ muốn thế nào muốn chụp như nào thì mình sẽ theo đấy. Thì theo thông tin mình biết thì sẽ có cái hybrid là trực tiếp bạn nào mà em không thì như là xong lại rồi, còn các bạn ngoài bạn nguyện vào được còn lại không thì mình nếu mình được thiết thì mình sẽ cho bạn lại và chuyển trường nữa. Thì mình đưa ra quyết định là sẽ rồi học vào 3 tiếng luôn để cho bạn không phải đi nhiều. Và quyết định thứ hai là cả hai buổi rồi còn như vậy còn một buổi nữa mình gộp thành hai buổi nữa thì hai buổi này mình sẽ để học ở trường và mình sẽ không rảnh và mọi những phần trăm là điểm đi đủ để cho giáo này một bạn nào mà ở tỉnh ngoài thì có thể là mở các bạn tham gia liên quan để học là như thế nào mà không muốn lên lớp thì là làm cho cái lịch như vậy. Ok. Okay, so do you have any question, comment? Right, so we will discuss on each other on the generalization and the declaration. So first, the variation is some formulation, right? So first, the generalization will affect one of proton in integration, and then the fusion formulation. This mutation spread out in the relation until it reaches a certain allen frequency will be considered as a variation. So first talking about numbers. So the human genome is considerably large compared to the other organisms in the world. So compared to the eukaryote, bacteria have the size just about 4 megabase. More or less 4 million base. The others memory have also similar genome size, about 3 gigabytes, and which a very popular analysis have extraordinarily large genome with a size of genome which is around 17 gigabytes. So we have discussed on the genome of prokaryotic cell and eukaryotic cell. So in eukaryotic cell, the large component of the genome is now coding protein. So they are like here intron and the intergenic gene. That means the regions. That means the region in between genes. Meanwhile, in the bacteria, let's say a large proportion of the genome is coding for protein, and they are organized as occurrent. And this was really clear when comparison between the nuclear genome of human and the mitochondrial genome of human. So the mitochondria is bacterial organism. So which means that when comparing between the nuclear genome and the mitochondrial genome, means that in the human nuclear genomes, they only 1.5% was coding for protein. And meanwhile, in the mitochondrial genomes, there's around the coding sequences take around 97 of the regions. Only 3% of the region. Only 7% is the deluge of regions is not coding for protein. Right. So in the human genomes, we see that only 1.5% is coding for protein. The intron is taking about 90%. The intergen is taking about 33%. And in the other, the last portion is replication. So replication is mostly focused on the telomere and in the female chromosomes, and also in the region the genome what called junction fragment genome. But replication have also have application in forensic science, and especially in the marker that we discussed at the end of the chapter. So let's go back a little bit on history. So in 1953, the first name model was discovered by two scientists, Watson and Crick. And in 1977, first sequencing method based on standard sequencing method was invented. This was based on the DNT, when it's in the side to the interface, it will start up the activity in the right there. And as a consequence, we will have different fragments of the DNA with more or less than each other, only one nucleotide. And from that we can identify the nucleotide at that position. In the same year, we have the first genome piece was sequenced with the genome is from bacteria with almost just like base. And in 2003, the most important achievement in the science of chemistry have been published. The first human genome was sequenced. So this is a synthesis of 100 labs and 1,000 scientists across the world. So at the moment when the first human genome was finished, the press, the media were talking about this a lot, that we have the code of life, code of God. We understand about we can enjoy human will, and so on. Have a better treatment for inheritance. But soon later, they realized that sequence the human genome is just a first step, a beginning step, which is simple and easy compared to the later step, which is much more difficult, much more time consuming, labor consuming, and money consuming. It is annotated the genomes. Or in other words, find out which sequence is a gene and what is functional of gene, and what is consequences if we have a mutation, and this mutation, that mutation, and what is the consequence if knocked out one gene. So since 2003, from the time from 2008 to 2015, 1,000 human genome project was launched in 2018 with the development of the new next generation sequencing. So it's second generation sequencing. Now in 2022 we have the third and the fourth generation of sequencing. So in 2018 there's 10 countries are completing sequence 100,000 of their citizens. Among those are US, UK, Japan, China, France, Australia, and also other countries. And in the end of last year, in Vietnam, we have been decade. Completing sequence more than 1,000 Vietnam. And if you want to watch their achievement, you go to so it's mass they talk. So you can see here, they have sequence more than 108 individual Vietnamese individual. Among those, half female. They have this more than 60,000 genes. Among those, 19, nearly 10,000 is coding for protein. Nearly 17,000 is long coding RNA, and so on. They are 49 million variants. The size of the genome is more than 160,000 gigabases, with the classified raw sequence genomes is 64 more than 64,000. So compare, if I separate the human genomes with the coverage about 48 sequences is around 400, 500 gigabases. So let's have a compare with the FMBR genome in human. So you see that in human here. Sorry, the next question. We can click on more information here. You see that here, the human genome at the moment, the size of the genome is around more than nearly 4.1 billion base pair. Okay. Why is the number in the media category 3 billion, not 1 billion? because in that case it's the old version. Okay, so every time the sequence was released, this number will change. Okay, so in 2003, when those was sequenced in 2003, people think that the human genome will estimate is around 3.2 billion base pair with coding gene is about 60,000 genes. But then they sequence more and more people, and they realize it's just a variant. So the human genome was decreased a little bit, just like changes. So now the new version is around 3.1 million base pair. Okay. And then coding genes is 20,465 total coding, compared with big data mass spectrometry total. So they say in 1008 individual they found, you can see here, nearly 20,000 total coding genes. But in either the world, this is the thing that the data from all over the world. The world of human genomes is contained around more than 20,000 genes. Coding genes. Not coding genes is 24,000. Long reading is 17,700, which here is 16,000. The personal genes, actually, this is not a gene. This is just a fragment of DNA which has sequence similar to a gene, but because lacking automotive region, lacking region, lacking and few other controlling regions. So those genes will be not translated. So it's actually not the protein coding gene, but it has a sequence similar to the coding gene. So for the time being, don't pay attention on this version of the genes. Okay. And they have around more than 700 million short variants. So in the last autumn, if you want more details, you can go on exploration. So at the moment, they have not let the personal data, the detailed data exist public yet. So scientists still cannot use this data to investigate any phenotype future to see that any populate to phenotype future like BMI index or type 2 diabetes and so on and on. But hope that in the future they can open this information. So talking about mutation, what's a mutation? So we have discussed in chapter 2 mutation that in the way when DNA sometimes make a mistake. So in for example in the virus that use DNA as incorporated material, the rate of mutation is from 1 million to 100 million base pair. That means in 1 million base pair to 100 million base pair, we will have one mutation. Okay. With the virus using the RNA as genetic material, the mutation rate will be 1,000 to 100,000 base pair mutation. So in human, without any DNA mechanism, the mutation rate is around 100,000 mutation nucleotide one mutation. However, in organisms like human, they have DNA mechanism, and it will detect and prepare the DNA mutation. And this process successfully in 99% of the cases. So it decrease dramatically decrease the mutation rate from 100 million base pair for one mutation to 10 billion base pair for one mutation. So with the human genome was about 3.11 base pair. That means it will take around 3.2 to 3.3 as cell generation to have a complete one DNA mutation. So first talking about evolution. I have that code here. Evolution or not evaluation. Okay. So we can see in here is the migrating free human. So we based on the evidence on fossil and that we take around the world. We know that human was appear on this planet in around 10 million years ago in Africa. Okay. And about 5,000 years ago, they started to migrating in different direction. One is direction is to the south to Australia. I inhabit in Australia about 50,000 years ago. One another go north into Europe and inhabit in Europe, located in Europe about 40,000 years ago. And the other direction is move around to Middle East, to China, and go into north to America around 16,000 years ago. So based on genetic mutation and evolution, people find out that the skin pigment of humans, of the people who located near the equator, are somewhat associated with UV light, the UV light at that season. So, for example, in the right panel here, you're seeing the right panel here, you're seeing the UV index of the world. So around the equator, in the red and orange region is the region that the UV is strongest. Meanwhile, in the higher altitude up to north, down now, UV light was weaker. And similarly with the skin pigment of people located in the regions here. So this is the average skin pigment of the population located there. So of course, with the low latitude nearby the equator, people can have darker skin. We can have advantage in preventing skin cancer and suicidal later. But the higher latitude up to north, the people tend to have more pale skin. We can have advantage in producing vitamin D.
And this paper was published on Science in 2017, based on different population in Africa. Okay, so they have studied different population in the middle of the equator, in Saharan deserts, in Ethiopia, in the Saharan deserts, compared to the people located in the high latitude in the north or in the south. And they find out when GWAS, genome-wide association study, called genome-wide association study. So this is technique beyond identifying the SNP or marker across genome and evaluate the SNP associated within a six marker with a certain phenotype trait, which in here is pigmented skin. So in here, after the investigator GWAS of the genome to skin pigment, they find out SNP code RS 1426554. It is really controlled, really associated with the pigment of the skin. So we can see here the SNP here have two alleles, G and A. And meanwhile, there are people in the population located nearby the equator. The allele is take a major percentage. The people who live in the north, which have weaker UV light condition, tend to have the A as a major allele. Okay. So let's practice this which I call this on SNP. Okay, so I will go on SNP. So I will type my SNP here. 1426554. Go. Wait a minute. Okay. So we can see here. So this SNP was located in chromosome 15 in the forward strand. Okay. It's a submarine. That means it's located in the coping region, and it caused alteration in the amino acid. Let's click on phenotype data to see beside this if any phenotype data is related to. Okay, so according to research, this is also associated with the BMI, the body mass index, the associated allele A. So that means that if you have the allele A, you can have weight BMI index. Okay. So this also associated with the eye color, the hair color, and of course skin pigment. Okay. So let's go back to the distribution of this statement overall.
click on population census. So we can see that this network was in overall. This network was distributed quite equally, with the G is 56% and A is 44%. However, in Africa, the G is majority when it takes 93%. Meanwhile, the A is take only 7% per element C population. The same in East Asian. In East Asian, that means China and Japan, so on, Korea, and so on. This alien was G is the majority, 99%, and the A is 1%. So that means that and in Europe, in the contrary result in Europe, the age is the alien major alien with 100%. That means all the Europe population, none of those carry LNG. The road here means Caucasian, means white. So that means that if we take into account only this East Asian is low African more than Europe. Just kidding, because in genetic background, we must take into account the whole genome, not only one. So for the mutations, that there are factors that can cause mutation. You can categorize those into physical mutations, that means radiation, like X-ray or UV ray. You can categorize those as chemical mutation, like in the laboratory genetic molecular biology, we have bromide, we have the phytopathone, we have the polyamide substances that can be demonstrated really easy to cause DNA mutation. So in the future, if you are working in a lab like those, it's helpful with those substances. Okay. So this is publication on human mutation in 2018 of the Institute of Genome Research Institute that located very nearby USGH, just 2 blocks away. So they have performed sequencing in sugar, participated in the world environment that have been exposed long-term exposure with dieting. So the dieting is made component with the Asian population that the US Army will use this agent in order to destroy the jungle in terms of outcome. And at the time, they have not any effect of that vaccine on the human and also the animal model. They quite the instance that the vaccine just have an effect on that. But this application comes so that the vaccine have its own effect on the carrier genetic background too. So for example, in here, when they analyze the trio, that means both father, mother, and offspring, they find out that the trio with the father who have long time exposure with that vaccine, or with the vaccine concentration in the blood is higher than normal,
they have found DNA mutation that a significant rate of DNA mutation compared to control group. So in here, one of the mutation, large mutation, is the inversion of chromosome 12 in the family with the history of a long time exposure to live. So this paper is one of the evidence of the pursuit against most company who export the Agent Orange during the Vietnam. But until now, this is not been yet. So some of you have some of you have been practicing with the data bank in the practical session. So this is central online inheritance in man, which is a database for the auto disorder, genetic disorder, and autogene that relate to genetic disorder in human that following the Mendelian rule. And so among those lots of significant diseases such as color blindness, this is just a mutation located in the X chromosomes. Crenshaw, I mean, diseases that cause the newborn child have to cry sound like a sound cat. It's actually a deletion on the chromosome number 5. Cystic fibrosis. So until now, we don't have we still don't have the effective treatment for this disorder. It's just one mutation that is just related to one nucleotide. The chromosome 7. Glycogen syndrome, a very popular syndrome where an employee more than two gender chromosomes. Sickle cell disease, very fatal disorder that also until now we don't have cure for this mutation. It's just a mutation in chromosome 11. So let's go further. One example in disorder, we have Autism syndrome. Also, antamino. So what is antamino? Polypeptide. We have discussed in the previous chapter. This is called series phenotype like common in manual features, defective in the intellectual development, and also cardiac problems. So this was discovered through related to a gene located in the X chromosome called the NNA genes. And very recently, researchers find out that there is another gene located in the autosomal chromosome called the NNA15 is also related to disease. So this is not like a single gene disorder, it's polygenic disorder. I mean, this is disorder controlled by two or more genes. So let's watch this disorder. I don't know if I should things now.
Work, we discovered and report a new genetic disease with mutations in a gene called NA15. And in this work, we found 32 unrelated families from throughout the world, And we showed that they have mutations in this N815 gene that basically decrease the ability to modify other proteins of their structure and function. And these individuals have intellectual disability as well as other physical anomalies like congenital heart defects. And this complements really nicely our previous work with something called Ogden syndrome, which had mutations in the same pathway inside the cell. And so the broader applications of the work are that these days we're able to identify multiple families, like on the order of 30 or more families with mutations in the same gene, and that really allows us to get at trying to understand the full spectrum of the expression of various different traits in multiple families. And then as we as a society begin to sequence millions of people, we will be much more easily able to interpret mutations that we find in any one person based on the previous work that has been done on those more severe diseases.
So come to genetic mutation, we can categorize genetic mutation. It follows the level of the mutation. At the high level, we have the chromosome mutation, where all the mutations related to the structure or the number of chromosomes. And in the down to DNA level, we have DNA mutation, which is just related to a fragment of DNA. So I have made a comparison between the DNA mutation and chromosome mutation here. So mostly DNA mutation is just endosomal sequence in the nucleotide sequence of a gene, and it's caused by the failure in DNA replication by DNA polymerase. You use some mutagenesis factor like UV or some chemical compound. So because it's involved in just a few nucleotides, it just affects to one or several genes. And so the influence of those mutations is lower, normally lower than chromosome mutation. And sometimes it can be corrected because of the DNA mechanism, DNA mechanisms. However, in chromosome mutation, we see that this is the MO due to the disturbance in the number of chromosomes or structure of chromosomes caused by the crossing failure in the crossing over meiosis, including certain chromosomes or fragment of chromosomes. And because the whole or the fragment of chromosomes was effected, so many genes was related. So so normally the effect of the chromosome mutation is more than the DNA mutation. Normally they are harder to correct the body correct the chromosome mutation compared to DNA mutation. DNA mutation have the genetic mechanism, but chromosome mutation we don't have such equivalent mutation at the same level. So first talking about DNA mutation. So we have normal sequence here. We have so first let's what was the more think about different type of DNA mutation.
The term mutation brings countless fantasies to our mind, right from the ability to fly high in the sky to that of diving deep into the oceans. We all love to have such fantastic abilities, but these are all fictitious. Such fantasies don't exist. So can we say that mutation also doesn't exist? Hold on, that's not true. Mutation does exist. But mutation doesn't lead to such drastic changes in us humans. We can't have mutations that give us extraordinary powers. Rather, most mutations give rise to concern. They result in undesirable changes from time to time. In these upcoming videos, we shall majorly talk about mutation, what causes them, and their types in general. Let's begin with the definition first. Mutation, in simple words, is defined as sudden and abrupt changes to the genetic material of organisms. Now let's break this down to understand it better. Sudden and abrupt changes mean a few changes that occur suddenly and are mostly unexpected. These changes are usually random and disruptive to the normal functioning of the cell. And where do these changes occur? They occur in the genetic material of the organism. That means in the DNA. And as a result, either the gene or the complete chromosome gets affected. So mutations are spontaneous changes that affect the DNA or the genes or sometimes the complete chromosomes. Now since these are the areas that get altered, mutations are categorized on the basis of them. The two major types of mutation are genetic mutation and chromosomal mutation. Let us see these types. Let's consider this small section of the DNA. Let's say that this part changes into another. Another possibility is that either this base gets deleted or an extra gets inserted. In either case, the outcome is that the sequence of genes will change. Technically, the reading frame will change. The ribosome assembly, including the tRNA, reads the genetic code in triplets. These triplets are called codons. Now, if there's any change in a single nucleotide, then the entire codon will change. Thus, the reading frame will change. And what will this result to? That's right. The altered gene will give rise to a different protein than the usual one. And there are chances that this defective protein turns out to be a harmful one. So any change in the DNA or in the genes can cause a shift in the reading frame. As a result, an altered protein is produced. This was the first type. Now let's zoom out the DNA structure. What do we see? Yes, we see a typical chromosomal structure. Now tell me, what will happen if there are any alterations in its structure? And what if the total number of chromosomes get altered? Let's find the answer in the next part. This is a representative karyotype for human. By now we know what a karyotype is. Can you try to give me some conclusions of it? You may say that it looks perfectly normal, but let me spot a flaw for you. Do you notice this set? This is the 21st chromosome set. Instead of having a normal pair of chromosomes, here we find a triplet. Now you may say that it may be good for the individual as it has an extra chromosome, but that's not the case. Any change in the number of chromosomes creates a lot of defects in the normal functioning of the body. On this note, let's understand chromosomal mutations. As the name says, chromosomal mutation means the mutation of chromosomes. And how exactly does it get mutated? It can happen in two ways. One way is when the structure of the chromosomes gets disrupted. The other is where the number of chromosomes are different than the usual in the organism. Let's have a look at each type. This is the structure of a typical chromosome. Now imagine this small part gets physically removed. This would result in the chromosomes in getting smaller as its segment is lost. This also means that if the segment had some important genes, then they will also get lost, which can result in severe abnormalities. Such type of a mutation is called deletion. This is where the part of the chromosome gets deleted. Now imagine this part of the chromosome gets duplicated. As a result, the chromosome's length increases. Needless to say, this will also result in some or the other abnormality in the individual. This type of mutation is called duplication. Now comes the next type. These are two different gene segments. Imagine they get flipped. Does the chromosome appear like this? This is called inversion. Do you think this type of mutation is harmful? It may or may not be harmful depending on which genes get flipped. Now let's take one more chromosome. Imagine a part of this chromosome gets removed and gets inserted in this chromosome. What will be the result? That's correct. Both the chromosomes will change drastically. This type of mutation is called as insertion. And now comes the last type. Instead of a segment getting inserted to another chromosome, let's imagine a situation where a segment from each chromosome gets interchanged. So this segment will get into this chromosome and vice versa. This kind of mutation also results in abnormalities as both the chromosomes get altered completely. Any idea what this type will be named as? Well, this type of mutation is called translocation. All these mutations resulting in the disruption of normal chromosomal structure are broadly capped under a single heading. They are categorized as chromosomal aberrations. Now this is about the mutations that involve changes in the structure of the chromosomes. What about the mutations that involve a change in the number of chromosomes? Let's understand that in the next part. This is a normal chromosome, while this is a disrupted one. When the structure of a chromosome gets altered, it's called chromosomal aberration. We have seen this. Now let's move on to the second type. It's the mutation that involves a change in the number of chromosomes. It's called as aneuploidy. In literal terms, aneuploidy means the presence of an abnormal number of chromosomes in a cell. So the karyotype of a person with trisomy 21 is an example of aneuploidy. The presence of one additional chromosome in the 21st set gives rise to a total of 47 chromosomes in a person. So instead of a normal 46 chromosomes, this person has an additional chromosome. To be precise, an additional autosome. This case of aneuploidy is called trisomy 21. This is because there are 3 chromosomes instead of 2 in the 21st set. The problem of trisomy 21 leads to a condition called Down syndrome. We will get into the details of these disorders in our higher grades. But for now, let's focus on understanding the chromosomal mutations. This aneuploidy involves an autosome. Now tell me, what if the number of sex chromosomes alters? So instead of XY, what if an individual has XXY? Is that even possible in the first place? Well, yes. There can be individuals with an additional X chromosome. This condition is called Klinefelter syndrome. Such individuals exhibit both masculine as well as feminine characteristics. Now, in the case of females, imagine that instead of addition, there is a deletion of an X chromosome. This would mean XO. So this individual will have only one X chromosome. This condition is called Turner syndrome. There are a few developmental abnormalities in such scenarios. These individuals have a total of 45 chromosomes only. These are a few examples of aneuploidy that involve autosomes and sex chromosomes. Now, before we conclude, tell me one thing. What causes these mutations? How can DNA or genes or entire chromosomes get altered? There are several factors named mutagens which lead to alterations. Some of them include radiation, chemicals, and even certain consumable items. To summarize, we have two major types of mutations. One which involves alterations in the DNA or the genes. These mutations result in a shift in the reading frame, resulting in defective protein synthesis. Alterations in the structure result in chromosomal aberrations. They can be categorized as deletion, duplication, inversion, insertion, and translocation. The other is where the number of chromosomes are different than the usual in the organism. It's called a nucleoidy. The changes can be in the autosomes as well as the sex chromosomes. Here, the total number of chromosomes in a cell are different from the usual. This was about mutations in general. To find more interesting videos, do not forget to subscribe to our channel.
Okay, so now I guess that we will have a small break, 5 minute break before going to the next part. Okay. Okay, so let's come back to our lecture. So, talking about type DNA mutation, so we have normal sequence here. We have 5 type mutation. We have first is insertion, which is the new nucleotide here was inserted into to the normal sequence. We have a deletion. One nucleotide here was deleted. Is A. So we have only CTG left. We have duplication. One two nucleotide due to error in the DNA replication was copy duplicated by the DNA polymerase. We have inversion. This kind of mutation is really rare compared to the other. Where a certain nucleotide was inverse and changing position of each other. And we have substitution or replacement where DNA, so for example, TG here. So for example, an A here was replaced by a new nucleotide T. So one example for point mutation is the sickle cell disease. So sickle cell disease is the disorder, inherited disorder in which the erythrocyte, or in the other word is red blood cell, is have a moon shape like this instead of the plate shape, which it can maintain its function to carry oxygen and carbon dioxide in out of the bloodstream. But in the moon shape like this, the efficiency carry oxygen and carbon dioxide will decrease. So in the my type, we have a combination between the healthy erythrocyte and sickle cell erythrocyte. The patient will be suffer from like erythrocyte. And in severe case, when a large portion of the red blood cell was have a moon shape, the carrier can be killed at home. And the reason leading to sickle cell disease is mutation, just point mutation or mutation related to one nucleotide. So the nucleotide here is the A. The pair AD here was replaced by T. And after a few sequence DNA replication, because the DNA replication is automatically user DNA template here as a template inside the new DNA strand, so it will add the A to position the T followed by the rule. This AT here legally sequences that are GAG, which is coding for glutamine, is now replaced by GUG, which is coding for valine. Okay? Because this is coding for a beta globin structure protein from hemoglobin, so it leading to the structure of the protein was collapsed. The shape of the red blood cell will be dramatically changed from the red blood cell to a half moon shape cell like this. And this variant is have ID for BRX234. So let's have a quick look on this SNP on the NCPL database. So we go to the NCPL. Famous. So as you can see here, this is a mismatch variant. When click onto the phenotype data, with a few seconds. So we can see that this SNP is related to anemia, to beta thalassemia, or the disease that related to the abnormal condition in the blood. It's also related to glomerular filtration in the kidney. And one more thing is really interesting is it was considering related to malaria. So why malaria? So let's go back and explore it in the population genetics. So when you can see in here, this tip is really rare in the four population in American, in Asian, East Asian and South Asian, and in Europe, but it's take a significantly different proportion in Africa, with the A allele that takes around 10%. So the reason of this is by natural selection. In African is the reason where malaria is take a pandemic of malaria. And meanwhile, the 334 meanwhile the 334 causing the sickle cells. It also protects the carrier with malaria, because the malaria parasite can only invade into the healthy red blood cell, but not in the sickle cell anemia. So, as the consequences, in the region that is not a pandemic of malaria, like America, like in Asia, like in European, the sickle cell have a very low percentage, since the carrier will be nearly die from birth. But in Africa, where malaria kills hundreds of thousands of newborn child every year, this is considered going to be an advantage, since this also provide protection against malaria. So sometimes it's in certain circumstances, certain environmental factor, a harmful factor can be a beneficial factor. So the frame shift mutation is just like the point mutation is affecting to only one or several mutations, but it's not like a substitution, but it's more like insertion or deletion. And because of insertion or deletion, a number not divide around to 3 nucleotide, it changes the read frame of the DNA sequences. So, for example, if we have a DNA sequence like profile, we have yes, so this is normal DNA sequences. Because it's as a consequences, IDC is coding for methionine, GA is coding for glutamine, GA is coding for alanine, and this is coding for glycogen. But since we have a deletion of the G here, we will have now the ATC is the same, because this is 5 prime and this is 3 prime. But the next codon AA now will have combined to the G of the next codon to form lysine. And CA now will have to combine with the C of the next codon CCT to form histidine. So as a consequence, you can see that the whole amino acid sequence from the point of mutation will change. And the more nearly the beginning of the gene, the point of mutation is, the more damage effect that this mutation can cause to the sequence of the amino acids, and so the 3D structure and function of protein. So next, let's come to the chromosomal mutation. So chromosome mutation can be divided to the structure chromosome mutation, which is including several parts, just like the DNA mutation. We have deletion, where a part of the chromosome was deleted out. For example, the part B here. We have duplication, where we have a duplication of this one. We have inversion, where part of the chromosome was changed the direction. So instead we have the genes order was ABCD. So now we have its inversion from the position to the B, D, E. And we have translocation, in which the part, the unhomologous region of chromosome was exchanged to each other. And note that in the translocation we have this different with the normal cross-link, because in the cross link it's a normal phenomenon where the homologous region of two sister chromosomes was exchanged during meiosis one. But in translocation we have homologous region of two chromosomes will change to each other, and sometimes it's not from the sister chromosome. The second type of chromosome mutation is number of chromosome mutation. So number chromosome can divide into aneuploidy, where the number of chromosomes is just related to one extra or one less pair or several pair of chromosomes. So for example, in here, we have an aneuploidy due to the failure in the separate of the double chromosome during meiosis one, or sometimes is non-disjunction. So as a consequence, this pair here divide into three pair of double chromosomes, and when during meiosis two, it will provide two lineage with the extra pair of chromosomes. Meanwhile, in the cell that lacking a double chromosome is provide a gamete with less one pair of chromosomes. And the second type of chromosome mutation is polyploidy, in which the carrier we carry the whole set of chromosome number, and this is really rare in human or in animal models. It's more frequent in plant models. So we have different types like triploid, where the whole chromosome, a whole set of chromosomes was added into the genome. Tetraploid, when two sets of chromosomes are added in each pair of chromosomes, and histology and so on. So let's go into a specific case, Down syndrome, for example. So in Down syndrome, we this is number of chromosomes mutation, of course, is aneuploidy, where in the 21 pair, the pair 21 chromosomes, we have one extra chromosome more. So this type chromosome 21 is the size of the chromosome 21 is relatively small compared to the other chromosomes, but it's in chromosome 21 we have many really important genes located in chromosome 21. So if a failure during the meiosis 2 occur and the other gamete might have only one chromosome or one chromosome 21, normally the carrier will be killed from birth because lacking so many important genes like this, the carrier can be survived. The other carrier who have an extra chromosome 21 having typical phenotypic features such as abnormal of skull, abnormal shape of skull, short hand, cardiac defect, and teeth problems, and let's say abnormal facial feature, plate facial, and normally the Down syndrome patient will also have to suffer from defective intellectual development. That's what is small about Down syndrome.
As you're probably well aware, our DNA is like this humongous blueprint of information on how to make a human. Usually this massive document is packaged up nicely into a storage bin called a chromosome. Actually, usually we have 46 chromosomes that we use to neatly organize all our information, depending on how you define organized. Each of the 46 chromosomes is actually part of a pair of chromosomes, since you get one from each parent, so 23 pairs. If you wanted to make another human, first you'd have to find someone that feels the same way, and then you both contribute half your chromosomes, so one from each pair, right? 50-50. Now, what if someone contributes one too many? Say dad contributes 23 and mom contributes 24. Is that possible? Yes, and it's the basis of one of the most common chromosomal disorders, Down syndrome. Someone with Down syndrome has 47 chromosomes instead of 46. Specifically, they have an extra copy of chromosome 21. So instead of 2, they have 3. So Down syndrome is also known as trisomy 21. In other words, 3 chromosome 21s. Alright, so in order to package up half the chromosomes into either a sperm cell or an egg cell, you actually start with a single cell that has 46 chromosomes. Let's just say that we're making an egg cell for the mother. I'm just going to show one pair of chromosomes, but remember that all 23 pairs do this. So the process of meiosis starts, which is what produces our sex cells, and the chromosomes replicate. And so now they're sort of shaped like an X. Even though that there are 2 copies of DNA here, we still say that it's one chromosome, since they're hooked together in the middle by this thing called a centromere. Okay, now the cell splits in 2 and pulls apart the paired chromosomes. So in each of these cells, you've now got 23 chromosomes. Now the 2 copies of the chromosomes get pulled apart, and the cell splits again, which means 4 cells, each still with 23 chromosomes. Now these are ready to pair up with a sperm cell from dad that has 23 chromosomes as well, totaling to 46 chromosomes, and voila, 9 months down the road you've got yourself a baby. Now, with Down syndrome, or trisomy 21, a process called non-disjunction accounts for about 95% of cases. Non-disjunction means the chromosomes don't split apart. If the chromosomes in this first step don't split apart, then one cell ends up with both chromosomes and the other gets none. Then the final result is 2 cells with an extra chromosome and 2 cells missing a chromosome. Non-disjunction can also happen in the second step, though. So first step goes great, and both cells have a chromosome. But if they don't split apart in the second step, then the final result is one cell with an extra chromosome, one cell missing a chromosome, and two with the right number of chromosomes. Now, if a sperm cell combines with any of these that have a duplicate of chromosome 21, then the combined cell will have one extra copy of chromosome 21. In other words, 3 chromosome 21s, or trisomy 21. In case you were wondering, the sperm could also combine with these cells that have a missing chromosome. If that's the case, then there would be a total of only 1 chromosome 21, and we will call that monosomy 21. In my example, we followed the egg cell from the mother, but this process could happen the opposite way, where the sperm starts out with too many or too few copies of chromosome 21. Apart from non-disjunction, Robertsonian translocation accounts for about 4% of trisomy 21 cases. Translocation, in this case, is a fancy way of saying move from one place to another. So a part of one chromosome moves and switches places with a part from another chromosome. In this case, the long arm of chromosome 21 translocates over to chromosome 14, and you end up with 2 hybrids, one with both long arms and one with both short arms. This little guy with the short arms carries just a little bit of usually non-essential genetic information, and is typically lost by the end of meiosis. So there are a few ways that this can go down. First, including the translocated chromosomes, they replicate, and now these could split into one with both normal chromosomes and one with the long guy and the short guy. In which case, after splitting again, you'd have 2 normal cells and 2 cells with a big guy, since remember we lost the little guy along the way. So now contribute the other parent's DNA, and you've got 2 normal cases, and then these 2 cases are called balanced carriers. And we say that it's balanced because you've still got both long arms, and so most of the genes are still here. Kind of like a 2-for-1 deal. Okay, now let's say that the normal chromosome 14 ends up with the short, and normal chromosome 21 with the long. Now you get 2 cells with the normal and long arm, and 2 cells with the normal and short arm, which remember is usually lost. So these have 1 extra chromosome 21, since the long arms carry most of the genetic material for both chromosomes 14 and 21, and these ones are missing chromosomes. Now combine these with the other parents again, and you have trisomy 21 here, and monosomy 21 here, since it's missing a chromosome 21. If this process was switched such that chromosome 14 ended up with the long chromosome first, then you'd ultimately end up with trisomy 14 and monosomy 14. So, of the 12 possibilities, 2 end up being trisomy 21, right? With Robertsonian translocation, one of these chromosomes needs to be chromosome 21, right? Even though the other is usually chromosome 14, it can actually be other chromosomes as well, like chromosome 22. Finally, about 1% of patients are mosaic, meaning that their cells are mixed, and some have 46 chromosomes and some have 47. How does that happen, though? Well, let's think about what happens after conception. So you've got this one cell called the zygote that needs to develop into a human, right? Since we aren't single-celled organisms, it has to divide over and over again, essentially producing every kind of cell in the body. Now, each of these divisions is called mitosis. Non-disjunction of chromosome 21 can also happen during mitosis, in which case you'd end up with one cell that has an extra chromosome 21 and one without. So one with 47 total chromosomes and one with 45. The cell with 45 chromosomes isn't able to survive, but the one with 47 does survive, and continues to replicate and produce more cells with 47 chromosomes. If this happens early on in development, then you can imagine that a lot of cells down the line can have 47 chromosomes, right? If it happens later on, then less cells will have 47 chromosomes. Mosaics can also happen when the zygote starts with trisomy 21, or 47 total chromosomes. In this case, non-disjunction would produce one cell with another extra chromosome 21. So this one has 4 chromosome 21s, totaling to 48 chromosomes. And this cell can't survive. The other cell, though, loses one of its 3 chromosome 21s. So now it only has 46 chromosomes, which happens to be the normal amount of chromosomes, right? And this is sort of like the opposite situation, because now, through a mistake, you've actually ended up with the right amount of chromosomes. So if it happens early on in development, you'll actually end up with more cells with the normal amount of chromosomes. If it's late in development, though, then less cells will have the normal 46 chromosomes. Having an extra chromosome 21 is one of the most common chromosomal disorders, and not surprisingly, it has an effect on almost every organ system in the body. And for that reason, there's a long list of common issues for those affected with Down syndrome. Here are some of the major ones: septal defects in the heart's chambers, usually between the two atria, duodenal atresia, increased risk of acute lymphoblastic leukemia, mental retardation and an increased risk of Alzheimer's, and sterility in males. There are several physical characteristics as well, like a simian crease in the hands, a gap between the first two toes, a flat facial profile, and epicanthal folds. Having an extra chromosome 21 is thought to affect these systems by an overexpression of genes on chromosome 21, since there's an extra copy contributing to gene expression, which changes the development of all the systems just listed. Now, a major risk factor for Down syndrome is maternal age, the age of the mother. Trisomy 21 happens about once in 1,500 births where the mother is younger than 20 years old. Contrast it to about 1 in 25 births when the mother is older than 45 years old. During pregnancy, some screening tests can be done, and certain markers suggest higher risk that a baby might have Down syndrome. Alpha-fetoprotein, or AFP, unconjugated estriol, or UE3, are on average lower when compared to unaffected pregnancies. And human chorionic gonadotropin, or HCG, as well as inhibin A are both typically elevated compared to unaffected pregnancies. An imaging test using ultrasound can also be performed, where clinicians look for nuchal translucency. None of these are exact indicators of Down syndrome, however, but they do give valuable information about the overall risk of Down syndrome. Even though there's a long list of complications and hardships for people with Down syndrome, improved medical care has led to a much greater longevity and quality of life. Many individuals with Down syndrome are able to thrive and lead full lives. Thanks for watching. You can help support us by
So, one interesting information of the clip, we can see that the ratio of the risk ratio of getting Down syndrome is really associated with the maternal age. That means the younger the mother have the child, the less possibility the child getting from Down syndrome. So at the moment, there's a service provide even here in Vietnam. There's a service called when a woman doesn't want to have a child at the moment, she can choose to store her age at the age bank, where they can store the age of the woman in liquid nitrogen, to waiting for a more appropriate time for her to decide to have a baby. So another syndrome related to the aneuploidy is a Turner syndrome, where the aneuploidy happen in the gender chromosomes, which in here is a lack of one sex chromosome. So I know that in a normal carrier, female, we have 2X. Even in a normal carrier, we have 2X. One of the chromosome X was inactivated. We will discuss more on this phenomenon on chapter 7, why one of the X was silent. It's due to the DNA methylation to silence the whole chromosome X. But somehow, even if with the X was silent, the Turner will have also the different, significant different phenotype compared to the normal female. So the typical phenotype feature could be a short stature with abnormal development of the heart, poor breast development, the defective in growing the fertilization organelles, and normally the Turner syndrome patient will have not the ability to produce gametes. So another case is the Klinefelter syndrome, while the carrier have 2 chromosome X and 1 chromosome Y. So with this type of syndrome, the carrier will have a tall stature, but poor muscle development, and defective in fertility organelles, and defective also in the intellectual development. So on the triple X syndrome, where the aneuploidy also happens in the gender chromosome, but with 3X. So he or she will have a tall stature, but low defective in the development of the fertilization organ. So to sum up, the type of mutation, in mutation we can divide it in 2 types: at gene level, the DNA mutation. We have 5 types: insertion, deletion, duplication, inversion, and substitution. In the chromosome mutation, we have 2 types. First is the structure of chromosome mutation. We have also deletion, duplication, inversion, insertion, and translocation. We have chromosome number mutation, where an aneuploidy, where we have an extra or less number of one pair of chromosomes, and polyploidy, when we have a whole extra set of chromosomes. Okay. So I guess today's session will be finished here. Do you have any question or comment? The next part on evolution, we will discuss on it on Monday. Do you have any questions for the moment?
So is there a case of 2n plus 1 minus 1 and aneuploidy?
Yeah. We have also minus 1. We have also plus 2, minus 2, whatever. Yeah.
There are like 2n plus 1 minus 1. Does that exist?
I'm sorry, what? 2n
plus 1 minus 1.
Yeah.
No,
but normally the minus 1 will be have a less chance to survive more than the 2n plus 1.
I mean that both 2n plus 1 and 2n minus 1 combined. The chromosome count still remains 46, but there are three on one cluster and only one on another.
Well, I think it depends on the proportion of the fertilization. So, for example, we have an egg, we have a sperm here. Okay. So, if the eggs were normal, but the sperm was n plus 1, and this one is combined with the eggs, we will have 2n plus 1. But in another chain, if we have the sperm with n minus 1 combined with the eggs, we will have the 2n minus 1. So it depends on the ratio, which sperm is swimming quicker. It's based on statistical. Okay. Such a question, or do you want to ask about something else? Okay. So in case you have no more question, comment, I thank you very much for your attendance, and I wish to see you face to face in Monday. Okay. Thank you very much.